Welcome to the Deep Dive. We are here to give you the ultimate shortcut to mastery, extracting core knowledge from the most complex scientific sources. Today, we are opening the master blueprint, the operating manual for life itself, molecular biology.
That's right. And for you, the learner, this isn't just an academic exercise. This is the bedrock. Molecular biology is the study of the fundamental biochemical processes governing cellular behavior. Everything from, specialized function and cell interactions to the essential process of cell turnover.
Our mission is laser-focused. We want you to gain the foundational knowledge of molecular biology necessary to understand complex disease processes, specifically within otolaryngology, up to an elite professional standard. We are unpacking the mechanisms of life and showing exactly how their disruption creates pathology, which is, at the end of the day, the definition of disease.
And this foundational understanding is, well, it's non-negotiable for the modern physician. When you truly grasp these molecular mechanisms, you stop just seeing symptoms and you start seeing points of failure. This allows you to predict disease behavior, classify disease more accurately than conventional pathology, and most critically, explore methods for restoring cellular regulation at the molecular level.
So it's the path from broad treatment to highly rational, targeted intervention.
Exactly. That's the goal.
Okay, let's unpack this blueprint, starting with the structure of the genome itself. Hereditary information in eukaryotes is stored in double-stranded deoxyribonucleic acid,
Known universally as DNA, and that iconic structure is, of course, the double helix.
Right.
The helix is structurally stable because it's held together by highly specific hydrogen bonds between complementary nucleotide pairs. And you know the rules that govern all of life. Adenine, or A, pairs only with thymine, or T, in cytosine. C pairs only with guanine, or G.
And this pairing isn't just a structural detail, is it? It's much more fundamental than that.
Oh, absolutely. It's the basis for high-fidelity replication and really nearly every single major molecular biology technique we're going to discuss later on.
And when the cell reads these instructions, it reads them in chunks. The nucleotides are organized linearly in triplets called codons. Each specific sequence determines a single specific amino acid, for example, the codon ACU, that specifically codes for threonine.
And what's fascinating here is that the system has redundancy built right in. You can almost think of it as nature's brilliant failure protection plan. There are 64 possible triplet combinations for only 20 commonly encountered amino acids.
So if the system is so specific, why the extra capacity? Why all the extra combinations?
Well, because several amino acids are represented by multiple codons. Take lysine, for example. It is specified by both AAA and AG.
Oh, okay.
This redundancy offers a crucial bit of protection against simple base substitution errors. If a small error occurs in that third position, there's a pretty good chance the resulting protein will still be the correct one.
So it lessens the likelihood of a catastrophic mutation.
Exactly. But not all 64 possibilities code for something. 3 of those codons are absolutely critical signals: UAA, UGA, and UAG.
These are the stop codons.
The mandatory stop codons. They signal the arrest of translation, ensuring the polypeptide chain stops building at precisely the right length.
Now that we understand the code, let's look at its geography.
Yeah.
The overwhelming majority, about 99.9% of all DNA exists in the cell nucleus.
That's the nuclear genome. In humans, this is estimated to be about 3,000 megabase pairs in size, which encodes a truly massive number of instructions, somewhere between 30,000 and 35,000 genes.
But we can't ignore the smaller sort of independent sibling, the mitochondrial genome, or mtDNA.
No, you can't. It's vastly smaller, only 16.6 kilobase pairs, and it encodes just 37 genes. And the structure itself contrasts sharply with nuclear DNA. While the nuclear genome forms that complex double helix and highly organized chromosomes, mtDNA is circular double-stranded DNA. It lacks the conventional chromosome structure.
But the inheritance pattern is the real biological differentiator.
Right. It really is. The nuclear genome, as we know, is inherited equally from both the mother and the father.
Okay.
But the mitochondrial genome is entirely maternally inherited. And while it was historically overlooked, we are seeing increasing recognition of the role of mtDNA mutations in human disease.
Especially in certain tissues.
Especially those involving highly energy-dependent tissues, like, certain forms of hearing loss or muscular issues. The field of mitochondrial genetics is just rapidly expanding because those 37 genes are so vital.
It's incredible to think about the logistics. How does 3,000 megabase pairs of nuclear DNA, which if you stretched it out would be over 6 feet long, How does that fit inside a microscopic nucleus?
Expert packaging. That's the only answer. DNA is packaged into chromosomes via complex folding, wrapping tightly around structural proteins.
And in humans, we have 23 pairs of chromosomes.
Right. Diploid human cells contain 22 pairs of autosomes, which are just labeled 1 through 22, plus 1 pair of sex chromosomes, either XX or XY. And each chromosome has a distinct morphology.
And when you look at a chromosome under a microscope, it looks like it's pinched in the middle by a central constriction. That's the centromere.
Yeah.
And that constriction separates the chromosome into two arms. The long arm, which is designated Q, and the short arm, designated P.
And the centromere's role is absolutely critical. It acts as the anchor that attaches the chromosome to the mitotic spindle, ensuring proper segregation, that is, the correct equal division of genetic material during cell division.
So without proper centromere function, you get errors.
You get aneuploidy, which is a hallmark of many, many cancers.
We also have the specialized structures at the very ends of the chromosomes, the telomeres. They contain these simple repetitive sequences.
Their fundamental function is really dual. First, they maintain the structural integrity of the chromosome, so they prevent them from fusing together or being degraded. And second, they solve the technical problem of complete replication right at the extreme ends of the DNA strand.
So to navigate this massive genetic map, we rely on a standard nomenclature for locus assignment. It's a conventional naming structure, right? Understood globally.
That's right. It consists of the chromosome number, followed by the arm, and then the position on that arm.
So if a paper references locus 3p21, you immediately know you are looking at position 21 or 21 on the short arm, the P arm of chromosome 3. It's like a precision address system.
It is. So moving from structure to action, we have DNA replication, which is necessary for every single cell division. And this process is called semi-conservative replication, and it must be achieved with breathtaking fidelity.
So walk us through that process. What is happening during cell division?
Okay, so first, the two strands of the double helix have to be separated. That's the job of an enzyme called DNA helicase. It just unwinds the helix. Once they're separated, each single strand acts as a template for the polymerization of a new complementary strand. And the synthesis is catalyzed by another highly complex enzyme, DNA polymerase.
And the term semi-conservative defines the final product, right?
Exactly. Each new daughter DNA molecule consists of one original or old strand and one newly synthesized or new strand. That combination of half old, half new is why we call it semi-conservative. It's a perfect mechanism for preserving the genetic sequence.
And the accuracy here is truly one of the most remarkable things about biology.
Yeah.
The estimated error rate is less than 1 in 1 billion nucleotide additions. It's almost perfect.
That near perfection is vital. Remember, a permanent change in that DNA mutation can inactivate a gene essential for the cell to function or, control its growth. That high fidelity is achieved through a three-pronged defense system.
What are those three mechanisms?
Okay, so first, the replication process is strictly unidirectional. It runs 5' to 3'. Second, the DNA polymerase complex has rigorous proofreading capability built right in. It actively detects mismatched DNA bases and corrects them on the fly. And third, if an error does slip through, we have highly efficient backup DNA repair pathways that patrol the genome, excising and correcting damage after replication is complete.
That sounds like an utterly robust system, which makes the failure so clinically significant. What happens when this system breaks down?
The failure of these high-fidelity mechanisms results in inherited diseases that are characterized by genomic instability and a very, very high incidence of malignancy. We see this classic pattern in inherited conditions like xeroderma pigmentosum, Fanconi's anemia, and ataxia telangiectasia. The breakdown in their specific DNA repair pathways just leads to the rapid accumulation of replication errors and mutations.
And that manifests as cancer.
It manifests as cancer.
Let's shift now to what a gene actually is and how it's turned into a functional product. We defined a gene as a region of chromosomal DNA that ultimately produces a functional ribonucleic acid, or RNA molecule.
And that gene is complex. It's composed of 3 key elements. First, you have the regulatory DNA sequences, which act as the on-off switch, determining when and in which cell types the gene is expressed.
Okay.
Second, the coding sequences, called exons. Those are the parts we read. And third, interspersed within those exons are the non-coding DNA sequences, or introns.
Here is the first real surprise for people new to the field. The human nuclear genome contains 3 billion base pairs, but only about 10% of that is actually coding DNA.
Yeah, that's right.
The remaining 90% is non-coding, or what was once, I think, pretty dismissively called junk DNA.
Yes, and within this vast non-coding expanse, you find short arrays of repeat units, often or trinucleotides, known as microsatellite repeats. And while their precise global function is still being mapped, their existence and their high dispersion have been enormously useful.
They act as molecular landmarks. Right.
Exactly. They facilitate the study of inherited disorders, and as we'll discuss later in segment 4, they are critical markers for analyzing tumors using the concept of loss of heterozygosity, or LOH.
Now, let's get that code to work. It starts with transcription. This is the intranuclear process driven by RNA polymerase, which uses one DNA strand as a template to synthesize a single complementary RNA strand.
And the key chemical difference is that in RNA, uracil or U replaces thymine T. The resulting primary RNA transcript then immediately undergoes post-transcriptional processing, which is often called splicing. The traditional belief was pretty simple. Splicing just removed the non-coding intronic sequences to produce the mature messenger RNA, or mRNA.
But here's where the complexity multiplier kicks in: alternative splicing.
Correct. We now understand that one gene can actually result in the production of several different, though often related, proteins in different tissues. The splicing machinery can choose which exons to include or exclude, effectively making multiple protein versions from the same original gene template.
So that vastly increases the functional output from a relatively limited count of 30,000 to 35,000 genes.
Enormously. Once that mature mRNA is finalized, it exits the nucleus and enters the cytoplasm for translation. It acts as the template for polypeptide synthesis, a process that's regulated and catalyzed by cytoplasmic ribosomes.
And this is where transfer RNA or tRNA comes in.
tRNA molecules recognize the mRNA triplet code and sequentially add the corresponding amino acid to the growing chain. The process starts with the initiation codon, usually AUG, which specifies methionine, and is terminated by those crucial stop codons we mentioned.
That growing chain isn't finished yet, though.
No, not at all. Finally, the polypeptide undergoes variable degrees of post-translational modification. This might include folding, chemical modification, or cleavage. This final step is what produces the mature functional protein product, which then performs its role either inside the cell or, if it's a secreted protein, is exported outside the cell.
This is where the story gets really current. The classic DNA to protein pathway is just the main highway. In the last decade or so, we realized that gene expression is meticulously fine-tuned by classes of non-coding RNA molecules.
Yes.
This dramatically complicates the picture. This is where that junk DNA narrative completely flips on its head.
Precisely. These non-coding RNAs are absolutely essential. We first encountered small interfering RNAs, or siRNAs, in research settings because they are such effective tools to deliberately suppress gene expression.
Okay. But the natural counterparts are the microRNAs, or miRNAs.
Yes, these are small, non-coding RNA molecules that regulate gene expression by base pairing with specific messenger RNAs, preventing them from being translated into proteins. It's like a regulatory mechanism putting a very precise molecular brake on certain messages.
And the scale of this fine-tuning is immense.
It's estimated the human genome contains well over 1,000 microRNAs, each potentially regulating dozens of different mRNA targets.
This regulatory layer has direct and immediate implications for our field. You mentioned their association with otolaryngological disease.
Yes, certain microRNAs are strongly associated with oral cancer, and what makes them particularly valuable is that they are secreted into body fluids, such as saliva.
That's a game changer.
It really is. They become extremely promising candidates for non-invasive disease detection, allowing us to screen for cancer markers simply through fluid analysis without needing invasive biopsies.
Beyond the microRNAs, we also have the long non-coding RNAs, or LNCRNAs. This is a relatively new and vast frontier of study.
LNCRNAs are proving to be masters of regulation. They're involved in nearly every aspect of gene control: transcription, splicing, translation, and especially epigenetic regulation. They can act as scaffolds, guides, or decoys to control complex cellular processes.
So these molecules, which we once ignored, are now implicated in major pathologies.
Absolutely. Although the field is new, LNCRNAs have already been implicated in major diseases like cancer and the processes of aging. This just underscores the point we made earlier, that 90% of the genome once discussed is actually complex, dynamic, regulatory machinery that we are only just beginning to decipher.
So if DNA is the blueprint, these tools are the forensic kit we use to read the crime scene and understand how the blueprint failed. To start, if DNA is so stable, how do we ever cut it up to study it?
We use restriction endonucleases, the molecular scissors. DNA is incredibly stable, and if you want to study or clone specific sequences, fragmentation is necessary. The first enzyme, Hindi pronounced Hindi 2, was identified in a strain of Haemophilus influenza way back in 1970.
And in nature, these enzymes act as a bacterial immune system, right? Recognizing and cleaving non-self DNA, like from invading bacteriophages.
Exactly. It's a protection mechanism we've cleverly repurposed. We now have a massive array of these enzymes sourced from various bacteria. Each one recognizes a highly specific target site, a sequence of 4 to 8 nucleotides. And the length of that recognition site is crucial because it dictates fragment size.
Give us an example of that statistical difference.
Sure, an enzyme that recognizes a 7-nucleotide sequence, a heptanucleotide, will statistically cut much less frequently than one recognizing a 4-nucleotide sequence, a tetranucleotide. So consequently, the heptanucleotide cutter yields much larger, less frequent fragments, which is exactly what you want if you are trying to clone a large gene sequence.
And when these enzymes cleave double-stranded DNA, they don't all cut the same way. We get 2 major types of ends.
Correct. The most useful are the cohesive or sticky ends. This happens when the cut is staggered, leaving short, single-stranded overhangs like those produced by Equer. Sticky ends are highly efficient for joining DNA fragments back together, a process we call ligation.
Which is the molecular glue.
Molecular glue. The alternative is a cut that goes straight across both strands, like that produced by SMA. These result in blunt ends. You can ligate blunt ends, but the process is far less efficient, making sticky ends the preference for, constructing recombinant DNA.
Once we have cut the DNA into fragments, we need to sort them out by size. That's the job of electrophoresis.
Right. And it works because DNA fragments carry a net negative charge due to the phosphate groups on their backbone. So when an electric current is applied across a gel matrix, the fragments migrate toward the positive electrode.
And it's a race based on size.
Entirely. Smaller fragments navigate the gel matrix more quickly than larger fragments. And we determine the size of an unknown fragment by running a graduated DNA ladder alongside it, which contains fragments of known molecular weights.
And the choice of gel matrix dictates the resolution.
Absolutely. For ultra-fine resolution, say resolving differences of just a single base pair, which you need for DNA sequencing, you must use polyacrylamide gels. They have extremely small pore sizes.
For the vast majority of routine work, like resolving fragments from 100 base pairs up to 20 kilobase pairs, we use agarose gels.
And for the absolute largest fragments, those exceeding 50 kilobase pairs like large bacterial artificial chromosomes or BICs, you need specialized equipment. This requires pulsed field electrophoresis, which periodically changes the direction of the electric field to force those huge molecules to migrate.
Moving on to detection, we use hybridization. This is the concept of specific annealing, the joining of a single strand of DNA or RNA, which we call the probe, to a complementary DNA sequence in our sample, the target.
And this simple concept allows us to detect the presence of a specific sequence. We achieve recognition by labeling the probe, either radioactively, which we localize via autoradiography, or more commonly now, with fluorochromes, which fluoresce when excited by light.
This brings us to the famous blotting family of techniques. The original Southern blotting, named after scientist Edwin Southern in 1975, involves transferring electrophorous DNA from an agarose gel onto a nitrocellulose membrane.
Right, which creates a stable, permanent record for hybridization. Then came the jargon terms. Northern blotting is exactly the same process, but applied to the transfer and analysis of RNA.
And Western blotting is used for protein analysis. Here, electrophorous proteins are transferred from a polyacrylamide gel onto a membrane, but the detection mechanism changes.
It does. Instead of a nucleic acid probe, detection relies on specific antibodies that are labeled either radioactively, enzymatically, or fluorescently. So 3 different molecules analyze 3 different blots.
Shifting scale, we look at cytogenetics, the study of chromosomal abnormalities and rearrangements. This field really began when the correct human chromosome count was settled back in 1958.
And classical cytogenetics is still crucial for prenatal diagnosis, think trisomy 21 and Down syndrome, and for identifying specific translocations in blood cancers, like the chronic myeloid leukemia translocation T922.
However, using classical methods in solid tumors like head and neck cancers is often hampered.
It is. It's difficult to establish robust short-term primary cultures, and you can get randomly acquired chromosomal changes in vitro that just muddy the waters. But despite these difficulties, early studies were vital. They identified consistent chromosomal breakpoint areas like 3p21 and 17p21, suggesting locations for tumor suppressor genes, and amplification sites like 3q and 8q, implying the presence of proto-eng genes.
To bypass these culture limitations, we developed fluorescence in situ hybridization, or FIH.
Yes, and this uses fluorescence to target specific DNA sequences within the intact nucleus. ASH is essential because it allows us to analyze the copy number of a known specific DNA sequence directly on solid tumor sections, like breast or head and neck tumors, without having to culture the cells first.
But the real power move that fused fluorescence and whole genome analysis is comparative genomic hybridization, or CGH.
This technique permits rapid, medium-resolution screening of the entire genome for gains or losses of DNA. The mechanism is conceptually beautiful. You take matched tumor DNA and normal patient DNA, label them with two different colored fluorochromes, say red for tumor, green for normal, and then comparably hybridize them onto normal metaphase chromosome preparations.
What does that look like under the microscope?
Under red-green dual-filter microscopy with computer analysis, the interpretation is clear. If a segment of DNA is balanced genetic neutrality, it appears yellow. If the tumor DNA is underrepresented, meaning there's a loss of DNA, it appears green. And a loss often suggests the location of a tumor suppressor gene.
And if the tumor DNA is overrepresented?
Then you see red. Overrepresentation or amplification suggests the location of an oncogene. CGH was a revolution because it immediately refined the location of chromosomal aberrations in solid tumors, obviating the need for those difficult short-term primary cultures.
Now let's turn to the technology that literally changed the world of molecular biology overnight. The polymerase chain reaction, or PCR. This is the amplification revolution.
It really was. The original hurdle in the 1980s was getting enough of a specific DNA sequence for sequencing and cloning. PCR solves this by providing faithful and exponential amplification. The only prerequisite is that you must know the sequences flanking the DNA stretch you want to amplify so you can design specific oligonucleotide primers.
Let's walk through the cycle. It relies on precise temperature control. It's 3 segments, repeated 30 or more times.
Segment 1 is denaturation. You raise the temperature sharply, usually to 94 degrees Celsius for about 30 seconds. This disrupts the hydrogen bonds, effectively separating the double-stranded DNA into 2 single strands.
Segment 2 is annealing, or hybridization. The temperatures drop substantially, typically to 55 to 65 degrees Celsius.
Right, and that allows the specific oligonucleotide primers to hybridize or stick to the denatured DNA strands.
And segment 3 is extension.
Extension. The temperature is raised again, usually to 72 degrees Celsius. This is the optimal temperature for the DNA polymerase to extend the strand, incorporating the deoxynucleoside triphosphates to build the new complementary strand.
The power is what's staggering. Each cycle doubles the number of DNA copies. If you run a typical 30-cycle PCR program, you amplify a single copy of a DNA sequence. What, 268 million-fold?
An astounding 268 million-fold. It is massive power, and that power created a massive problem for the initial inventors, Mullis and Faluna. The DNA polymerase they used was denatured cooked, essentially during that 94-degree denaturation segment.
So they had to manually replace it after every single cycle.
Every single cycle. It was laborious, expensive, and just impractical for widespread use. The genius solution came in 1989 when Lawyer isolated and characterized Taq polymerase.
From a very unusual source.
A very unusual source. It came from Thermus aquaticus, a thermophilic bacterium that naturally lives in hot springs. Because the bacterium lives at high temperatures, its DNA polymerase is naturally heat resistant and doesn't denature. You just set it and forget it.
But that immense power, that 268 million-fold amplification, is a double-edged sword. It's also its Achilles heel, right?
Absolutely. The sensitivity is so high that PCR is incredibly prone to contamination. Even tiny amounts of stray DNA, perhaps from the investigator's skin or from dust in the lab, can be amplified exponentially, leading to false or artifactual results. Rigorous clean room practices are essential.
There's also the issue of fidelity.
Okay.
The original Taq polymerase lacks that proofreading function we discussed earlier.
Right. It's fast and heat resistant, but somewhat sloppy. For high-accuracy applications like sequencing, newer cloned enzymes, such as food polymerase, are used because they incorporate that crucial proofreading function, significantly increasing the fidelity of the amplification.
And we have a specific challenge in solid tumor analysis, particularly with squamous cell carcinomas of the head and neck. or HNSCC.
The problem is in situ contamination. These tumors have a large stromal element benign surrounding tissue, so a total DNA extraction contains a mix of malignant tumor DNA and normal stromal DNA, and that contamination can mask the true, subtle genetic alterations within the tumor cells themselves.
To eliminate that noise, we use laser capture microdissection, or LCM.
LCM is a fantastic solution. A laser pulse selectively lifts specific, pure tumor cells, sometimes down to just 30 micrometers in diameter, onto a film. This guarantees that we are analyzing only the malignant DNA, giving us a clean molecular signal.
Let's transition now to how these perfect molecular mechanisms break down and initiate disease, focusing specifically on cancer. We have to start with the foundational concept of tumor suppressor genetics, Alfred Knudsen's two-hit hypothesis.
This hypothesis, proposed in 1971 based on his statistical analysis of the childhood cancer retinoblastoma, was a monumental landmark. It established that two genetic events are required to fully inactivate a tumor suppressor gene, which is the gene mitigating against cancer.
And he noted the clear distinction between hereditary and sporadic cases.
Yes. In hereditary retinoblastoma, the patient already inherits one mutated copy of the RB gene in every single cell. Therefore, only a single secondary somatic event is needed in one retinal cell to initiate the tumor.
Whereas in sporadic cases,
you need two separate somatic events to occur sequentially in the same cell. This two-hit requirement really set the paradigm for cancer genetics.
It did.
And Webster-Cavaney later provided the molecular proof, showing that neoplasms arise in a multi-step manner, that chromosomal events are drivers of tumors, and that the physical loss or reduplication of chromosomes can lead to the expression of recessive mutations.
This brings us directly to the concept that proves the two-hit hypothesis. Laws of heterozygosity, or LOH. If you have one good copy of a gene, the wild type, and one bad copy, the mutant, the simplest way to reveal that bad copy is to just delete the corresponding wild type allele.
And that deletion results in homozygosity at that locus. So the inference we draw is this. Areas of the genome where tumors frequently show allelic loss are prime locations for putative tumor suppressor genes.
And LOH detection relies on those molecular landmarks we mentioned earlier. The highly polymorphic non-coding microsatellites, the short tandem repeat units.
Critically, these repeat units vary in length between the two alleles in a heterozygous person. So if a person is heterozygous for a specific marker, PCR amplification yields two different length products on a gel.
So we amplify the DNA from the tumor, and if it only yields one product,
then the tumor shows loss of heterozygosity. This implies the allelic loss of the other chromosome segment, the one that held the wild-type gene. This is the molecular evidence for the second hit. It's important to note, though, that individuals who are homozygous for a marker are non-informative for LOH testing at that specific site.
Cavaney's work also elegantly detailed the 6 different chromosomal mechanisms that could achieve this state of LOH. Let's walk through those 6 possibilities slowly, because this is crucial to understanding cancer genetics.
Okay, we'll start with the most obvious. Mechanism 1 is deletion, the physical loss of the wild-type allele, leaving only the mutant allele. Mechanism 2 is non-disjunction and loss. The entire chromosome carrying the wild-type allele is simply lost during mitosis.
Okay, 2 down. Mechanism 3 is non-disjunction and reduplication.
Right. So the wild-type chromosome is lost, but then the remaining chromosome, which carries the mutant allele, duplicates itself, resulting in 2 copies of the mutation.
Then number 4, mitotic recombination.
An exchange of segments occurs between the homologous chromosomes, resulting in a portion of the chromosome having 2 mutant copies. Mechanism 5 is gene conversion. The mutant sequence is copied over the wild-type sequence using the cell's repair machinery.
And mechanism 6, which is the quiet outlier, the simple point mutation.
Yes, a secondary mutation occurs in the remaining wild-type copy. And the key distinction here is that if we are using a microsatellite marker to detect LOH, 5 of those 6 mechanisms will result in the loss of 1 PCR band. But that quiet little point mutation mechanism F completely escapes detection.
That's a critical point. The overall chromosomal structure remains heterozygous when only a point mutation occurs. So a tumor that looks genetically normal by LOH analysis might still have 2 inactivated copies of the gene.
Exactly. You must confirm LOH results with sequencing to catch those point mutations.
By analyzing the extent of allelic deletion across the genome, a process called allelotyping, we identify common areas of loss linked to specific cancers.
Yeah.
For head and neck, squamous cell carcinoma, or HNSCC.
For HNSCC, common LOH areas are consistently observed at chromosome 9p21, 17p13, 13q14, and regions on 3p and 8p. These loci represent some of the most frequently inactivated tumor suppressor genes in this cancer.
So we've established that gene inactivation requires two hits. But what about converting a protein-coded gene, a normal cell growth gene, into an active oncogene?
Oncogene activation usually only requires a single hit. This conversion can happen via gene amplification, which results in massive overproduction of the normal protein, driving uncontrolled growth. Or alternatively, it can happen via point mutations that affect the control of protein activity, such as locking the protein into its permanently active state. We see that in key players like CRBB2 or KRAS.
Let's quickly classify the types of mutations we see. Insertions and deletions of nucleotides are relatively rare in coding DNA, but if they occur, they're usually catastrophic.
Yes, because they cause a reading frame shift that dramatically alters all subsequent codons and thus the resulting protein sequence. It's usually a total loss of function.
Base substitution is far more common. This single base change can lead to three possible outcomes.
Right. Loss of function, typical of tumor suppressor genes like P53 or RB. Gain of function, typical of oncogenes, or no functional effect at all.
That's the silent substitution, thanks to what's called third base wobble.
Because multiple codons often specify the same amino acid, remember our Valiant example, GUA, GAC, GUG, and GU all encode valin. Substituting that third base often results in no change to the final protein.
But the substitution can also result in a nonsense mutation.
Right. Here, the base change creates a stop codon prematurely. This leads to the truncation of the polypeptide chain, resulting in a protein that is dramatically reduced in function or just completely non-functional.
Beyond changes to the DNA sequence, genes can also be inactivated by transcriptional silencing. This mechanism is purely epigenetic.
Epigenetics involves altering gene expression without changing the underlying DNA sequence. Transcriptional silencing often occurs through the methylation status of short DNA stretches known as CpG islands, which surround promoter regions.
And methylation is like placing a sticky note on the promoter, just switching the gene off.
That's a perfect analogy, and this mechanism is highly specific. Housekeeping genes are unmethylated in all cells, ensuring they are always active, but cell-specific genes are only unmethylated and thus expressed in the relevant tissue.
And the clinical revelation here
Is that many classic tumor suppressor genes like CDKN2A, P69NK4A, APC, and RSF1A are now recognized to be inactivated not by dilution or mutation in many cancers, but by hypermethylation of their promoter region in malignant tumors. The cell is turning off its own self-defense mechanisms.
To truly map this complexity, we've moved beyond single gene analysis into the omics world. Let's start with next-generation sequencing.
The current strategy is incredibly efficient. You construct a library of fragmented DNA, then use biotinylated RNA baits to specifically capture the exomic sequences, the functional coding parts, and isolate them using streptavidin-coated magnetic beads. This is followed by PCR and massive parallel sequencing.
And this strategy achieves incredible depth. For HNSCC, this high-throughput sequencing revealed the entire mutational landscape.
It did, including the frequent, previously unidentified inactivating mutation of the NOTCH1 gene, illustrating that the genetic drivers are often more complex than we initially thought.
Then there is proteomics, the global profiling of proteins in cells and serum. Methods include two-dimensional or 2D gel analysis, X-ray crystallography, and mass spectrometry.
Right.
Why do we need to analyze proteins if we already have the DNA sequence?
Because proteomics gives us information that genetic profiling misses entirely. Post-translational modifications. Proteins are constantly being changed after they are built. Modifications like phosphorylation or glycosylation, and some of these are cancer cell specific. Proteomics allows us to compare the entire pattern of functional proteins between tumor and normal tissue, giving us a functional view analogous to the genomic view provided by CGH.
And finally, metabolomics, the global analysis of small molecules, the metabolites. This gives us the ultimate picture of what the cell is actually doing right now.
It is the newest discipline and the most challenging because no single technique can analyze all metabolite types simultaneously. It requires a sophisticated mix of gas chromatography, high-pressure liquid chromatography, and capillary electrophoresis to separate the molecules, followed by identification via mass spectrometry.
And the clinical application is already here.
It is. Metabolomics has been successfully applied to detecting oral cancer via the analysis of specific small molecules accumulating in saliva and serum. The breakdown products of cellular function can serve as non-invasive biomarkers.
Let's conclude this segment with the ultimate aberration, cellular immortality, which is controlled by telomere biology.
Right. Telomerase is the enzyme that maintains telomere length in germline cells, sperm, and egg cells, but it is generally absent in normal somatic cells, like your skin or liver cells.
And this absence gives rise to the theory of aging.
Correct. The progressive shortening of telomeres with each cell division eventually leads to a critical point where the cell enters senescence or stops dividing, which contributes to the aging process.
The cancer paradox is that some cells survive this normal aging endpoint.
Yes. Critical telomere shortening can cause genomic instability, leading to what's called a cell crisis. However, a small proportion of cells that survive this crisis will then activate telomerase activity. This leads to cell immortalization by maintaining telomere length indefinitely, making uncontrolled growth possible.
And the clinical data here is striking. Telomerase activity has been demonstrated in 80% of oral cancers and about 50% of oral leukoplaskia lesions.
And its activity is directly associated with the degree of dysplasia. This suggests that the activation of telomerase is not an initial event, but occurs during the late critical stage of oral premalignancy, turning a dysfunctional cell into a potentially immortal cancer cell.
So that mechanism provides a clear target.
A very clear target for anti-telomerase drugs, especially for hitting persistent, highly resistant cancer stem cells.
Moving into the translational aspect, let's look at how we locate disease genes, especially when we don't have an abnormal protein to study. The traditional approach, used for conditions like sickle cell anemia, was forward genetics. Find the protein first, then find the gene.
But that approach fails when the protein is rare, inaccessible. Imagine trying to find a protein deep within the cochlea or simply unknown. This necessitated the modern strategy: reverse genetics or positional cloning. We determine the chromosomal location first, then isolate and sequence the gene to deduce the protein structure afterward.
Strategies for determining these chromosome locations include classical cytogenetic analysis, molecular analysis using LOH studies, and linkage analysis.
Linkage analysis is particularly effective when you have large family pedigrees that clearly demonstrate an inherited disease pattern. It relies on the construction of a high-resolution linkage map, which uses genetic markers spaced less than 1 centimorgan apart across the genome.
And we determine linkage by analyzing the co-inheritance of the known genetic marker and the disease trait through generations. This statistical probability is expressed as the LOD score.
Right, which stands for logarithm to the base 10 of the odds. A critical threshold is used here. An LOD score of 3.0 or higher statistically favors linkage. It means the odds are 1,000 to 1 that the marker and the disease gene are physically close.
And a score of minus 2.0
Effectively rules out linkage.
Once that linkage is established, those genetic markers flanking the disease gene are used as signposts to identify large chunks of genomic DNA, which are cloned into yeast artificial chromosomes, or YACs, or bacterial artificial chromosomes known as BACs.
And these are organized into a physical map of the region, allowing researchers to home in on candidate genes. Candidate gene identification is significantly accelerated by comparing the region to known animal models, or by characterizing which genes are specifically expressed in the relevant tissue. For example, when mapping hereditary hearing loss, you would specifically look for genes expressed in the cochlea.
Let's discuss the challenge of creating a progression model for HNSCC. The adenocarcinoma sequence in colorectal cancer gave us a beautifully rigorous, step-by-step roadmap for malignant progression. Are we ever going to get that kind of clean clarity in head and neck cancer?
That's the challenge. The heterogeneity of head and neck carcinomas makes defining an equally rigorous universal model very difficult. However, we have identified key frequent events. The inactivation of the tumor suppressor genes P16 and P53 are two of the most frequent and critical molecular events in head and neck carcinogenesis.
And understanding that sequence allows us to move into risk prediction. That landmark study by Rosen et al., analyzing LOH in oral dysplastic lesions, provided an astonishingly clear risk stratification tool.
They found that LOH at chromosome 3p and or 9p occurred in virtually all cases that progressed to frank malignancy. This implies that genes at these loci act as essential gatekeepers, and their loss imparts a 3.8-fold increased relative risk of progression.
And the risk stratification gets even more powerful.
It does. If the lesion showed additional LOH at other specific sites, 4q, 8p, 11q, or 17p, the relative risk for developing cancer skyrocketed to threefold. This is predictive power far, far beyond traditional histopathology, allowing clinicians to intervene much earlier and more aggressively.
To process this level of genomic information quickly, we rely on gene array technology, or gene chips.
Gene chips allow for the rapid assessment of the genetic profile of cells or tumors. Up to 30,000 genes can have their complementary DNA or cDNA placed onto a glass slide. Hybridization with a DNA sample yields genomic information. But if you hybridize with an RNA sample, you get a semi-quantitative analysis of gene expression.
Which gives you a dynamic view of cellular activity.
Exactly. And as we learn more, we develop designer chips, which are tailored for screening known panels of mutations associated with specific tumor types. Furthermore, array technology has been combined with CGH, creating array-based comparative genomic hybridization, or ACGH, which completely removes the need for those labor-intensive metaphase chromosome spreads.
So if this molecular revolution is happening so quickly, what does this translation from laboratory to bedside look like for the clinician right now?
It affects three areas: classification, prognosis, and treatment. In classification, molecular analysis has already provided the clarity needed to differentiate diseases like neurofibromatosis type I from type 2. We anticipate similar clarity for syndromic and non-syndromic sensorineural hearing loss.
That clarity allows for better patient management.
Precisely. It allows clinicians to predict progression and target monitoring more appropriately. For example, identifying the specific gene defect might allow for the use of miniature infusion pumps to deliver targeted agents directly to the inner ear, circumventing systemic barriers.
And prognosis and staging are also being refined.
Yes, molecular characterization of tumors should eventually provide a more accurate prognosis than histopathology alone, aiding rational treatment planning. Research is actively defining genotype-phenotype correlations for resistance mechanisms, such as radio and chemoresistance. If we know a tumor carries a mutation that makes it inherently resistant to cisplatin, we can skip that line of therapy altogether.
And molecular assessment is already being used to refine staging, though technical complexity is a hurdle.
Elegant demonstrations have shown its power in assessing resection margins and in detecting subclinical metastatic disease in regional lymph nodes using highly sensitive molecular markers. While technical difficulty currently limits widespread adoption, the future of precision surgery will rely on this.
But the simpler, non-invasive techniques are coming fast.
They are. The emergence of molecular markers, miRNAs, metabolites, and circulating tumor DNA in saliva and serum will soon play a key role in the non-invasive surveillance and follow-up protocols for head and neck cancers, providing a powerful, accessible tool for recurrence monitoring.
Finally, let's discuss novel targeted therapeutics, where we exploit the genetic abnormality itself. The best example of this rational design is the therapeutic use of ONYX015.
ONYX015 is a beautifully engineered therapeutic virus. It's an E1b55 kilodalton gene-deleted adenovirus. The key is that it was designed to selectively replicate in and lyse cells that are deficient in the p53 tumor suppressor gene, while critically sparing normal, healthy cells that have functional p53.
That's molecular precision targeting.
It is. This tumor targeting property led to successful phase 2 trials when ONYX015 was combined with standard chemotherapy agents, cisplatin and 5-fluorouracil, in recurrent HNSCC. Post-treatment biopsies confirmed the virus's replication selectivity in vivo, demonstrating the immense promise of molecular biology in designing therapeutic agents that are active only where the pathology lies.
We have completed a comprehensive journey today, starting with the hydrogen bonds of the double helix, moving through the revolutionary forensic tools like PCR and CGH, applying the rigorous logic of the two-hit hypothesis, and glimpsing the future of omics technology and truly targeted therapeutics like ONYX015.
This foundational and detailed knowledge of molecular biology is no longer specialized. It is essential for the clinician. We've shown how the disruption of regulation leads to disease, and conversely, how understanding that exact mechanism allows us to predict, classify, and potentially restore function at the cellular level.
But here is the final provocative thought to leave you with, one that brings us back to that massive, enigmatic 90% of the genome. Molecular biology has revealed that what we thought was noise, what we call junk DNA, is actually complex critical regulatory fine-tuning, managed by microRNAs, long non-coding RNAs, and complex epigenetic methylation patterns.
So consider this. How much of disease, even in well-studied pathologies like HNSCC or complex conditions like sensorineural hearing loss, do we still fundamentally misunderstand simply because we have not yet fully mastered the intricate regulatory language of the non-coding genome? That, the full decoding of the vast regulatory darkness, remains the true next frontier for elite translational research.